As often as we possibly can, we like to gather and talk about the science that's underway aboard the International Space Station. For over a year now, we have been in the utilization phase, and there's a lot of science going on. And we figured what better time than on the eve of the launch of the SpaceX Falcon 9 and Dragon on its way to the International Space Station to restore America's capability to take cargo up and bring it back down from American soil. This will be a great time to get together and talk to you about science. So we've gathered a panel, and I'd like to introduce them right now. To my left is Julie Robinson, the International Space Station Program Scientist for NASA. Next to Julie, Tim Yateman, Interim Chief Scientist for the Center for the Advancement of Science in Space, or CASIS. Next to Tim, Sheila Nielsen-Price, cell biologist from Montana State University. And next to Sheila is Scott Smith, a NASA nutritionist from the Johnson Space Center in Houston. Thank you all very much for being here. We'll open up with comments, and then we'll be happy to take questions from the gathered press. Go ahead, Julie.
Well, thanks a lot. It's great to be here today on what is really a momentous milestone for research on the International Space Station as we head towards our first commercial cargo launch. The SpaceX Dragon is a really important vehicle for us because it supports the laboratory use of ISS both in bringing cargo up to the space station and in bringing research samples home. And it has a great return capability. It essentially replaces that capacity that we lost when the shuttle retired, so that now we'll be able to bring home a wide variety of biological samples, physical sciences samples, and we'll be able to bring home research equipment that we need to refurbish and then relaunch again for the next set of experiments. So we're really excited to be here and to share with you some of the science both that's going up and down on the flight, as well as give you some background about the research going on the space station today. At any one time, and now is no exception, we have over 200 experiments active on the ISS, and about 100 of those right now are US-based experiments, and the others are from our international partners, Russia, the Canadian Space Agency, the European Space Agency, and the Japanese Aerospace Exploration Agency. This SpaceX launch for the entire partnership will be taking up about 1,000 pounds of research cargo, equipment samples, in addition to the things that it launches for the crew, as well as for the vehicle maintenance and operations. And it will be returning about the same. And so this is a pretty exciting thing for us. The other excitement that we have is in seeing the development and the growth of our national laboratory users. And so it's exciting to have scientists from CASIS representing CASIS today to talk about some of the activities going on with CASIS on the national laboratory side. We have a combination of both NASA and national laboratory users. And to give you a sense of what that looks like, if I could have my first slide. Here in this graph, you see the different disciplines that are on the International Space Station in this research portfolio I've been talking about: biology and biotechnology, earth and space science, education and cultural activities, human research, physical sciences, and technology development and demonstration. And in this little pie chart, I show you those 100 U.S. investigations, but they're broken out in two ways. The part of the pie on the right that's all crunched together shows you the NASA-sponsored investigations, where NASA's the primary funder. The blown up part of the chart that's on the right side of the pie are those managed under ISS as a national laboratory. As you probably recall, back in 2005, Congress designated ISS as a national laboratory, and that's for users from other government agencies, the private sector, or nonprofit organizations. This increment, we have about 40% of the U.S. research that is on that national laboratory side, primarily because there are a large number of student experiments going up on this particular launch. The student education program has a wide variety of experiments going up, and you may have seen a press release about that just yesterday. The research, as I showed you then on that graph, there's a real diversity of research, and I wanted to give you just a sense of the kinds of things going on the space station today. So if I could have my next chart, I'll give you one example from each of these disciplines. In the physical sciences, we have a wide variety of experiments. Just one of these is a combustion experiment where we're burning solid materials. Now, in space, you don't have convection, meaning warm air does not rise, and that really changes the way that things burn. Scientists are leveraging that both to understand understand combustion better to make more efficient engines, but also to understand combustion better in case there is a fire on the spacecraft. And this study is particularly focused on solids that are in spacecraft that might catch fire if you had a big accident, and how you would suppress that burning by understanding that burning better. And you can see some pictures. This experiment has been ongoing for a while. You can see some pictures of things burning inside the microgravity sciences glove box. I think it's pretty cool that there are things burning like that on the ISS at different points in time. And what we're bringing up on this flight are igniters, the little thing that we use to help set these samples on fire. If I could have the next chart, switching gears to the Earth and space sciences, this instrument is on ISS and will be becoming more and more active as we continue to implement it. It's an instrument called ISERV, which stands for the Environment Research and Visualization System. It's basically a telescope with a high-resolution camera. It can take images of the Earth with 3-meter spatial resolution, so very high-quality, high spatial resolution images. It is linked to a project that's supported by USAID, the Agency for International Development, and to a project called SERVIR, which helps to distribute data of major environmental events all around the world. So things like droughts and floods and other kinds of dynamic events that can really have humanitarian impacts. So this telescope will basically be able to point out our optical quality window and collect data and provide it to this SERVIR network. It's a pathfinder instrument for us to help us see how we can get this kind of high resolution data. It'll be the highest resolution data we've collected from ISS so far. But its real importance is for supporting the environmental decision making around the world, and as a pathfinder for the major external instruments that we're in the process of building right now. Another area of research that we do on the International Space Station is technology development and demonstration. And by doing these kinds of demonstration activities, we can make sure that the next space exploration that we do is safer and more cost-effective. One example of a piece of hardware on ISS today that's important is the MISSY-8 experiment. And if I could have the next slide to show a picture of that. MISSY-8, here you can see an astronaut on EVA right next to the MISSY-8 hardware. This is a system for testing materials on ISS, and some of the commercial providers of satellites have found that if they test a new material in this way, they really learn things about those materials. The space environment is quite harsh. It gets hot and cold. There's ultraviolet light. There's atomic oxygen in low Earth orbit that can really chew things up. And so by testing these materials, satellite providers can have better performance and can take new advanced materials that are developed and be sure they're going to work before they put them in a spacecraft. And that really saves development time. Then shifting gears yet again to another discipline, we have human research that's really focused on understanding the effects of space on the human body so that astronauts can explore beyond Earth orbit. And in the next picture, you'll see an example of this, a photograph of Mike Fossum and Ron Guerin a few months ago putting samples into our Melfi freezer. That's a minus 80-degree laboratory freezer that we have on the space station. They're putting in samples of blood and urine that they had collected to monitor their physiological status. And one experiment that they've been sampling for is an experiment called VASCULAR, which is short for Cardiovascular Health Consequences of Long-Duration Spaceflight. It's a Canadian Space Agency investigation, and it's focused on, during spaceflight, whether the blood vessels change, if they look more like blood vessels of older people, and if there are inflammation of the artery walls, as well as other changes in the vessel properties. And that links to the loss of cardiovascular fitness that can happen to astronauts because it's so easy to float around in space rather than really having to fight gravity every day like we do here on Earth. So this is just one example of a number of human research investigations that are active on ISS today. Of course, if the human body is so affected by being in space, it's not surprising that cells, animals, small plants, and so forth are also affected. And so I wanted to give you one example of an experiment in biology and biotechnology. The plant signaling investigation, it will have samples coming home on this flight. And if I could have the next picture, you can see a picture of Sonny Williams actually carrying out operations on this investigation. On the next chart, please. So this is a study that's looking at the effect of microgravity on the growth of plants, and especially the way the plant uses plant hormones to signal and coordinate the growth of the different kinds of cells that it has. This is important both for food and crop production on Earth, as well as possibly understanding better how to produce food plants in space. And if I could have the next picture, please. Next picture. This final picture shows you an example of an educational activity. This is the Tech EdSat. It was deployed from ISS on Thursday. It's an example of an engineering education activity. It was built by students at San Jose State University. It has a ham radio transmitter in it to communicate with it, and it's about a 4-inch cube. The entire experiment was built for about $30,000 by using good commercial components. And one of the things that we're seeing in many of our education projects is we've brought the cost of doing research down enough on ISS that it's really accessible for students to do small tests and different things. If you noticed on that on the pie chart I showed, there was a really large wedge of education projects on the National Lab side, and that's driven by about 23 to 26 investigations that are student designed. There have been some contests under the Student Spaceflight Experiments program, and the winners of those contests, those lucky schools, are getting to fly their experiments. Each experiment is in what we call a mixed stick. It's basically a little stick. It's sort of like the stick that you use, the glow sticks that kids use at night or at concerts or things like that, and you break them to mix the fluids together. The astronauts will take them out, break them to activate the experiment, and then they'll be returned home. About 7,000 students have applied, developed experiment proposals for the SSEP program, and so that has a really amazing impact. Scientists or educators have studied middle school and high school students and find if they get to do real science where they're generating their own hypotheses, that's what really engages them in STEM education, in science, technology, engineering, and math. So we're really excited for those students, and in particular we had some experiments that were not successfully activated on a previous flight, and they're all getting to refly this time. And one of the great things about SpaceX Cargo and having these frequent launches is that when something does go wrong, which it does in the laboratory, we're able to recover it very quickly. So that kind of gives you a sense overall of the diversity of activities, the number of activities on the space station. The last year of full utilization from the time that the shuttle retired until today has been just extraordinary. We've had record numbers of crew hours going to research. We've had record numbers of investigators active on ISS. And so with that as context, let me hand off to Dr. Tim Yateman, and he can give you some updates on CASES.
Sure, thank you, Julie. Well, as you may know, CASES stands for the Center for Administration of Science and Space, and basically our organization is responsible for partnering with NASA to really identify the best science to go up to the station, to the National Lab. And we've been spending our first year not only developing the organization, but starting to actively look for both solicited and unsolicited projects to go up in space. In fact, projects that will unfold during increment 33 and 34 will be 3. One is called Spheres, which is a robotics programming competition where satellites inside the International Space Station will be programmed. It's a software competition for students to test out their capabilities. There's another program called Windows on Earth, which is basically, again, a software program to basically take advantage of Google Earth Engine and provide better visualization capabilities. And a third is called ISAC, the International Space Station Agricultural Camera, which captures some of these fantastic geographical areas of interest with red and near-infrared band passes and medium and high spatial resolution. So that's what's going on now, but in the future, what we're trying to do is identify the best science to go to the station. And so what we've done initially is we assembled what I would call a blue ribbon panel of scientists and reviewed basically what's been done to date to try to identify key areas of interest. We've identified really 3 main pathways, one in the life sciences, material sciences, and then Earth observation. And I can tell you a lot more about the life sciences where we made some significant progress. We've actually identified within life sciences about 4 areas of interest. The first is protein crystallography. And you might put up a video behind me. If you're not familiar with this, protein crystallography is the beginning of drug discovery for big pharma. So most of the drugs that are made today, the targeted drugs, particularly the targeted drugs, which are coming out of the Human Genome Project, start with understanding the protein structure. And the better you understand the protein structure, the better you can make a drug. And it turns out that there is a promise, a significant promise, that proteins can be better crystallized in the microgravity environment than on Earth. And so we're now going to really test that out. And one of CASES's goals is to engage commercial interest. And so that would be big pharma. And so you'll see on the CASES website some announcements with big pharma interest in projects in space. The second area of interest we found was in looking at osteoporosis. Osteoporosis is a significant disease affecting postmenopausal women in particular, and aging women. And so it turns out in space, astronauts are significantly affected by bone loss when they come back from the microgravity environment. And so it appears to be a very good recapitulation of the human problem. And so we're now looking into potentially putting mouse models up in space that could be tested with drugs or wild type and transgenic mice, comparing proper controls for these mice to really study and potentially develop drugs for osteoporosis. A third is muscle wasting. Again, another problem that came from the astronauts in their post-trip evaluations. We found they lost muscle mass, and that exercise, alone wasn't sufficient to prevent that problem. It turns out that the muscle loss, again, appears to recapitulate some of the human capacities we see where there's muscle loss, the human diseases where we see muscle loss. And so again, there's a pharma interest in potentially developing drugs to target muscle wasting. And we're also looking at other problems such as immune loss, which is a problem, and the potential for stem cells to be grown better in space than on Earth. We've had a number of unsolicited proposals submitted, I think about 35 to date, and we're progressing to fund some of those. And we've also had our first solicited RFP, and that was in protein crystallography. And so I think we're progressing on a number of fronts, as well as material science. There's been an unsolicited proposal there that will likely be funded. So all in all, very exciting and a very diverse group of experiments. So thank you.
Okay. Well, let me start by saying this is our first flight opportunity, and myself and my whole team is thrilled to be part of it. We're actually looking at our experiments are called Micro 6, and we're interested in analyzing Candida albicans, which is a yeast, and how it responds to the microenvironment of microgravity. There are a few things that we're looking at. One is Candida albicans is a commensal organism. It's common, but it usually doesn't cause many problems. But it is also a human pathogen, an opportunistic human pathogen. So when our immune systems take a hit and aren't functioning as well, Candida albicans finds an opportunity to cause infection, and those infections can be things like thrush or fungal nails. So we think it's important in its own right because of that potential. Because Candida does have this infectious potential, the role of these experiments, really the data that we're trying to acquire, is dual in function. One is we really want to assess whether Candida albicans poses any health risk to the flight crew. And then we're also very interested in learning what we can about how Candida albicans responds to this environment and use that to extrapolate how we think it might be responding in different aspects of the human body. One of the aspects of microgravity that we think is influencing Candida albicans is low fluid shear. And if you'll go to the first slide, to try and demonstrate what does fluid shear really mean, if you think of a stone in a body of water, if that body of water happens to be a waterfall or a rushing river, there's going to be a lot of influence of the water flowing over, rushing over the stone to that stone. In contrast, and that's a high shear environment. In contrast, if you think about that stone in the bottom of a lake where there's very minimal shear, the influence of the water is going to be very different on the stone. We basically think about cells growing in liquid very similarly. If a cell is growing in an active liquid environment, it's going to experience a high shear impact on its cell membrane, and it's going to respond according to that pressure. If it's growing in a low shear environment, the response of that organism is going to be very different. If you'll go to the next chart, we can actually reproduce these different levels of shear in the laboratory. So in the top left panel, you'll see flasks of media. That's how we typically grow lots of organisms, including these yeast. And those are very rigorously shaking experiments, so that's an opportunity for high shear growth, if you will. And then on the right, really the only way that we can really get a microgravity and the low shear associated with microgravity is to send the bugs in space, which hopefully they're going to do tomorrow. So the hardware that we use, it's in that image, but I also have an example here. This is hardware that was developed by Bioserve, and what it does, our experiments are contained within these tricked out test tubes, if you will, so that there are 3 chambers, and the plunger is depressed on that to allow a sequential mixing of the reagents in 3 chambers. So on flight or on station, the plunger will be depressed to activate cell growth, and then the next plunger will be depressed to terminate growth. So all of that will happen on station before they come back at the end of the month. Low shear is, and I have alluded to it already, low shear is also probably an environment that the yeast might access in the human body. So for example, the nail bed is probably a location of low fluid shear, as are the brush borders and the kidney. So again, that's why we're looking at the response of the organism to this environment, because we think we can extrapolate what we find out there and bring it back and predict what might be happening in the human body. We're anticipating a lot of data from these experiments, and if you'll go to the next chart. One thing that we're clearly going to be doing a large molecular analysis. We want to know what genes are expressed, what molecular pathways are activated, and what molecular pathways are inactivated while in this microgravity environment. We're also going to be looking at a lot of physiological parameters. So what's illustrated here, just 2 different ways to look at the cells. Under high shear conditions, these yeast grow in the circular form, they look like balls up there. But when they're exposed to low shear, as we've done in the laboratory, we see an increase in the elongated filamentous form, which is associated, basically the combination of those 2 forms is associated with infectious organisms. The next slide, please. The next chart. Thank you. We'll also be looking at the ability of these organisms to form multicellular communities or biofilms. And again, this kind of structure is protective for the organisms when they encounter environmental stresses. So we'll be interested to see if there is an increase in the number of cells that actually make these communities. And the next chart, please. Something else that we found in our ground-based studies that was very interesting is on the right-hand side you'll see cells that are plated and they grow as a community. These are actually visual to the naked eye. They're pretty normal-looking, smooth colonies, but cells that were subjected to low shear make, on the left-hand panel, these very convoluted 3-dimensional structures. And so we'll be really anxious to see if we find cells that make those types of colonies when they're grown in the microgravity environment as well. Let's see where. So basically, the experiments. The other thing I was going to mention is we're also going to be looking at antimicrobial resistance. We think that's important because we are looking at an infectious agent. And so ultimately, the experiments that we're looking at are to assess the risk to the flight crew, but also to learn what we can about the molecular and physiological signaling that low shear environment and an overall microgravity environment produces in these cells, so that we can sort of take the next step in understanding what happens when these organisms are actually growing in special niches within the human body. Thanks.
Thank you, Sheila. Scott?
Excuse me. Well, I'm here to talk about everybody's favorite topic, blood and urine samples. We're very excited. Sorry, we're very excited about the SpaceX launch tomorrow. We're more excited for SpaceX to come home because what we're expecting is 384 syringes of urine and 112 tubes of blood to come home in this SpaceX Dragon capsule. These samples will support two of the experiments that my team has on board the International Space Station. The first one is what we call nutrition, simply enough. And that's a study looking at a broad-based biochemical set of parameters in blood and urine to try to better understand nutritional status and related physiological symptoms, things like bone metabolism, oxidative stress, you name it. We also have an experiment that these samples support known as ProK, and this is a countermeasure study where we're actually looking at using modifying the diet to help minimize using the diet to help minimize bone changes during flight. Now, to talk briefly about the way these samples are collected, I think the first picture shows Aki Hashide after one of his blood collections. The blood collections are just like they are when you go to the clinic here on Earth. We use a standard needle, standard tubes, and you can see he's smiling there, so it can't be all that bad. On the other side, one of the most common questions folks get when from NASA when you go out and talk is, how do people go to the bathroom? And while we do have a typical, what we call a waste collection system, or you might call a toilet, just like your toilet at home, it doesn't collect any samples. So when we want to collect samples for scientific reasons, we have to have a different way to do that. And what we do is we have what's called a urine collection device. And I brought one of these here that you can see. This is essentially a bag with this is the male version, with a condom cuff on the one hand. There is a different adapter for females. The crew member voids into the bag. Before flight, we put in a small amount of a volume marker, a chemical that goes in the bag. They void in the bag, mix it up, and then out the blue port in this end, they take one of these syringes and we'll fill this up with urine. And this is what comes home. And again, we're expecting just under 400 of these tubes of urine to come home. The other bag goes into another bag and then essentially goes into the trash. So while it may seem very strange to some folks, my typical line is that it may be to you, but it's gold to us, and that there's a lot of science that comes out of this. We've been doing these experiments for a number of years now. Actually, yesterday marked the 6th anniversary of our first blood collection on board the International Space Station. Mike Lopez-Alegria did that on Expedition 14. And we've published a number of our studies over the years. One of the more recent studies published last month in the Journal of Bone and Mineral Research. And what we showed there is that crew members that eat well, that have good vitamin D status, that exercise hard, can maintain their bone mineral density. And as I said, we published that last month. The image you're seeing there is the cover of the September Journal of Bone and Mineral Research. JBMR is considered the leading bone scientific journal out there, and we were honored to be selected not only to have our paper published there, but to make the cover as well. And while there are some nuances to the data that we found, that is, we've not yet entirely solved the bone problem, we have shown for the first time in 51 years of human spaceflight significant progress in maintaining bone mineral density. Again, there's some things that we still need to do in terms of understanding changes in bone strength. There's some things we need to do in terms of optimizing exercise, in terms of optimizing nutrition. But again, the fact that we're making progress in human subjects during spaceflight is very exciting stuff. And again, that's why we're very excited for just about 500 tubes of blood and urine to come home at the end of this month. I'll throw back to you, Mike.
Okay, thanks, Scott. Well, not only do we have members of the traditional media with us, but we are pleased to be joined by members of the NASA Social, who are using social media to report on the happenings around the SpaceX Commercial Resupply Services Mission 1. And we're going to take questions now. We'll start here with Marcia Dunn. Please state your name and affiliation and to whom you're addressing your question. Marcia?
Marcia Dunn, Associated Press for Dr. Smith. How old are these samples? I mean, I don't know if you've been stockpiling them ever since the shuttle stopped flying. I don't know if you brought any back in May. And how important is it to have the SpaceX capability to actually return things, unlike the other cargo ships?
Well, to answer your first question, no, we have not brought any samples back since the last shuttle flight. And one of the ways that when NASA knew the shuttle was going to retire, we actually flew extra freezers to the space station to hold those samples so that the crews could continue to collect samples on orbit, knowing that we would bring them back when we had a chance. And the novelty at this point of SpaceX is that this is the first real return vehicle for this type of samples. Obviously, we can get the crew home on the Soyuz, but the cargo capability of the Soyuz is extremely limited. So this is our first set of samples that have come back. So if you want to go back, I guess the oldest samples in those freezers has been since last July.
Are they all coming back in freezers? Is that?
That's correct. There's actually two different types of freezers we have. One is a typical powered freezer that's known as the Glacier, and that's where all the blood will be. We also have samples coming back in what's called a double cold bag, which is essentially a fancy lunch bag that we don't let them put their lunches in, but it has ice packs in there that after very intensive testing, we've shown that they can stay in there for 3 or 4 days, plenty of time from the point at which they come out of the freezer, go into the bag, and we get them back in our hands in the lab. And these are the same two technologies that we use to bring samples back on the shuttle. It's just now we've adapted them to this other vehicle.
And if I could, one question for Dr. Robinson. Yesterday there was an announcement about in 2015, 2 crew members spending a whole entire year aboard the space station. What does that mean to your team of scientists? How exciting is that? What can you really get out of it? And what have you learned from the 3 Russian experiences that went that distance years ago?
Sure. Yeah, that's a great question. We've been working across the partnership looking at this for a number of months. In the past, the Russian cosmonauts that have flown for 1 year, they flew at a time when both medical technology was not as advanced as it is today, and also when we didn't have the knowledge that we've gotten from the space station so far about exercise routines and nutrition, some of the things that Scott talked about. So today we're in a position where we think we know a lot more about what it takes to keep a crew member healthy for 6 months in orbit. But we know that for a variety of space missions that are under consideration, we really might need crew members to go a little bit longer. And so we can get a quick look at what we don't know by having a small number of subjects, like the astronaut and the cosmonaut that will do this one year duration flight. We can collect similar kinds of measurements on them as we do on our 6-month crew members, and it will give us just a couple of data points that will tell us, is the trajectory of what we see happening in the first 6 months, does that continue? Does it level out on a lot of these different parameters? And that's going to really change the way that we look at our long duration research program. So it will immediately feed back into our human research risk models and help us redefine where we need more information, where we're good to go.
James?
James Dean with Florida Today. Ms. Robinson, I don't think hardly any science flew on the demonstration flight back in May. Why not? And at this point, with this mission, does this represent a true full complement of this kind of science you would like to fly, or are you still sort of working up to what you would consider like your highest value cargo as they continue to demonstrate the ability to get up and down?
What we did in May, because it was a demonstration flight and there were some uncertainties, not so much about the success of the flight, but about the ground operations and things, with new teams handling samples, some of which have limited lives, things that are frozen and so forth. We primarily flew samples that we knew were degrading in orbit, and we brought those home on the demonstration flight, and we did launch cargo both ways. But it was, as you say, a slightly reduced complement from our normal full up. This flight is all out, maxed out, really filling our research needs, and this is what we're going to expect SpaceX flights to look like every time they launch in the future.
Thanks. And you talking about down mass, of course, compared to the shuttle. And did I understand correctly? Does Dragon actually, at least from a research perspective, equal what the shuttle could return in terms of capacity?
So it doesn't equal it mass for mass, but we've designed the space station so that we don't really need to bring up and down really large components. So for our research resupply, for keeping the lab running, it gives us everything our users need.
Mark Ratterman with Talking Space for Dr. Robinson. I saw a mention on the payload list of AMS cables. Is that for replacement or spares, or what can you tell us about AMS?
Yeah, so it's just a spare cable for one that we replaced on orbit.
And another question about iServe, and you mentioned it being the highest resolution device to fly on station for Earth imaging. How does that compare to other satellites and apparatus in orbit that's doing Earth imaging? Is it similar? Is it another step ahead?
So some of the commercial imagers that people buy imagery from, like OrbView, have about 1 meter spatial resolution. So it's not quite as high resolution as that, but for imagery that can be provided to the developing world for helping to solve problems, it's higher resolution. As a comparison, Landsat, which is the other sort of inexpensive data source that's often used as a workhorse around the world, that's 30 meter spatial resolution. So when you're trying to look at droughts or floods, that extra spatial resolution can be really valuable.
Hi, I'm Stacy Severn. I'm with the NASA Social. A question for Dr. Yateman. You mentioned that there was an unsolicited RFP that came in for protein crystallography. I'm just curious, do we see more of that in the future? Independent corporations wanting research done on the space station?
Well, we certainly hope so. I think by generating interest through solicitations, we obviously generate a lot of advertising for what can be done on the space station, and we hope that unsolicited will come through as well, and we have a mechanism to filter those and examine them. So,
Art Harmon, SaveManSpace.com. 2 quick questions. Why just 1,000 pounds of cargo up and down on this mission? Its capacity is a lot more.
Well, that's not the total cargo up and down, that's the research cargo up and down. So we also have food, clothing, spare parts, all kinds of other things. The flight is full.
Excellent. And a follow-up on the 1-year mission, I believe one of the highest values of the space station is simulating Mars and other deep space missions. Will there be additional, or is it just going to be this one shot in 2015 due to the Russian scheduling of the tourist?
Well, so I want to emphasize that this 1-year decision was made across the partnership for scientific and technology testing value. Mm-hmm. The plan is to do it once and then see what we identify. So, as I talked about, if we're seeing that most of the trends are exactly what we would predict, then we would know scientifically we don't really need additional 1-year missions. But if we see some big surprises come out of our observations of the 1-year crew member, that's going to send us in a different trajectory. So the great thing is, across the partnership, we have an active team that is working on all of these different mission simulations and what should we be doing to get all the knowledge we need from the ISS period so that we're ready to go on to those future destinations. And we'll be able to assess that as we go, as we learn from this first mission.
Excellent. Thank you.
We're going to take one more question here, and then we're going to take a question on the phone bridge, and then we'll come back. So let's take a question here.
Steve Carney with the Florida News Network. Dr. Yateman, you mentioned the possible collaboration between cases and Big Pharma. I was just curious as to what sort of interest you've generated so far, if any, and what sort of time frame you're looking at for a collaboration to get started and possibly see some effects from it.
Sure. So I think on the website, when an announcement was made that Merck has an interest in a particular protein crystallography experiment with a monoclonal antibody, which looks very exciting. I think that as we develop capabilities, further capabilities, for instance, mouse models to go in space, we'll be able to engage a number of different pharma for various things. One of them is muscle wasting diseases, as we talked about, and osteoporosis is another. We also think stem cells might be an area of interest for pharma as well. So I think the more we can develop and partner with NASA and partner with industry to figure out what they actually need, I think we can entice them. So we're going to build things that they actually can use, not that they might use.
Okay, let's go to the phone line and take a question from Tariq Malik. Tariq, are you there?
Yes, thank you very much. Tariq Malik from space.com. And I think I have a question that may be either for Dr. Robinson or Dr. Smith. I'm curious how the demonstration flight, what with the launch and then the return, gave you the experience on what to expect in handling the return samples, from a capsule like Dragon, on getting it back from the ocean, getting it back to Houston, and into the hands of your scientists, how that process went, in terms of smoothly, and then also just how important it is to really have maybe a more frequent schedule of samples returning back on a regular basis from either Dragon or in the future Cygnus as well. Thank you.
Sure. So, Tariq, this is Julie. The test went really well. It's actually not that difficult on the ground because what we can do is take the cold bags, these freezer bags, and throw them right in a freezer. That keeps them cold until we get the capsule, until SpaceX gets the capsules ashore, and then we can offload those and transport them back to the users. With the freezers, we plug them in, and we did a test of that to see how well things held temperature. And so we really felt confident that we could bring home these essentially priceless samples, and that we'll be able to do so routinely. It's been a great performance by SpaceX to help us get all of those logistics worked out. And the other great thing is, as we move towards these routine SpaceX flights, we're also working closely with them to add additional capabilities that we may need to support our research community. So, for example, because of the interest from pharma, we know that we need more freezer capacity than we had originally planned, and so we're working to extend that, and that will happen in a couple of flights. Also, as we add more animal capacity to do animal research in the program, we work closely with SpaceX to enable that.
Tariq, do you have a follow-up?
No, that answers my question. Thank you very much.
Okay, thank you. Back here at Kennedy Space Center.
Jeff Faust, the Space Review. For Dr. Yateman, can you give us a timeline for some of these future research opportunities? How soon will some of these possibilities in the life sciences and other areas actually be flying to the space station?
So I think that the timeline is, I'm going to give them the right increments. I think 37, 38 is we're talking about. So not too far away. It gives us enough time to get the RFPs out, sort through some of these experiments, find the best ones, and get them up there. Also prepare the equipment and do all the integration required to get the right equipment up there.
And for those of you who don't speak increment, that's a year away.
Okay. Over here.
Hi, I'm Charles Parker with Brevard Times, and also I'm the director of the Da Vinci Academy of Aerospace Technology at Merritt Island High School. And I wanted to talk, Dr. Robinson, Dr. Yeoman, talk a little bit more about the cost of CubeSats and any collaboration that high school academies, aerospace academies, can do to help us get in space.
CubeSat costs.
Yeah. So certainly the CubeSat example that I talked about today cost about $30,000. There are a number of CubeSat launch opportunities that have been enabled by something we call JSOD. It's basically a little launcher that's on the GEM module. So now what we can do is we can launch these CubeSats in excess capacity that we have at any time. We put them in this launcher. They go out through the GEM airlock on the Kibo module. They can go out. There's a robotic arm that puts them out into this launcher. Well, actually, the launcher comes in and they load it up, and then it goes out. And so it's relatively crew doesn't take a lot of crew effort, and so it's a great system to take advantage of that. And there have been a lot of different educational organizations that have been working on building CubeSats and have had trouble finding launch opportunities. So it's a pretty good capability to add for both education value, but also for science value. There are scientifically based CubeSats both being launched, that were launched this week, and being under development as well. And just to add for your students, I mean, the big change that's happened in the last year are the numbers of kinds of contests and the rigor with which the winners of those contests have developed their science. So I would definitely encourage anyone from the Aerospace Academy to keep up on those contests and start getting your students to apply.
As it's mentioned, that next year CASIS is going to support a 5-week middle school camp with 3 to 5 locations nationwide, and there's going to be a final middle school competition in August of 2013 for some of these activities.
Mark Ratterman with Talking Space. It's kind of hard to direct this question to one panel member because you've all touched on it, and I know it goes away from where we are right now with what we know and what research has brought us. But might there be an advantage to, in the future, designing spacecraft that would have some spin or something to generate a slight gravity component that would mitigate some of the need for the hard exercise, for the concerns with bone loss and muscle loss? Just an open question, please.
You know, we've had some teams at NASA that have looked at that pretty intensively. Without getting really technical, the simple way that I can explain it is it's a lot easier looking in 2001: A Space Odyssey than it is when you actually get down to the engineering. So there are 2 challenges with it. One is when you're spinning a system, all your fluid physics changes again because you're basically operating a centrifuge. So your thermal loops, your pumps, all your liquids, you have to design completely differently than we do today for space or than we do on Earth. So there's a set of engineering challenges. There's also a set of human challenges because when you're spinning, you get a vector that goes just one direction. If you turn your head a little bit, it's like being on an amusement park ride. So it's not the panacea for the human response either. In fact, it could be worse than microgravity pretty significantly. So that's a short answer.
Marcia?
Associated Press, for the meals going up, are those all American-style meals, or are there other partners who are contributing Japanese food, European, Canadian?
I didn't hear the first word.
The food going up. Is that all U.S. supplied food?
Well, we do our food as USOS food for the U.S. operating segment, and then Russian food typically launches on the Progress. And that does always include a mix of things provided across the partnership. But I don't know the specifics of exactly what's in these food.
And for Dr. Smith, you're a nutritionist, correct? Out of all the years that astronauts have been living on this space station full time, what have you learned about nutrition? Is there any particular diet that seems to be more conducive to a robust space life, coming back and all of that? I don't know whether you want to get any. I'd be just interested and curious to know what's working and what isn't working regarding food.
There's a lot of things we've learned. And, when I meet with the crews pre-flight, the first thing I tell them is, if I can only tell you one thing, it's that you need to eat. That getting enough calories, getting enough food, everything else will come along. All the vitamins and minerals come along with that. So that we're typically not cherry picking of, eat some more of this to get more vanadium. So energy consumption is the biggest thing, and that's, again, what we tend to drive home. And that is one of the things that was highlighted in the paper that just came out, that the crews that ate well and exercised hard, they maintained their body mass, meaning they didn't lose weight. They came back leaner. They came back with less fat, and came back again in pretty good shape. Beyond that, there's a number of things that we've learned either in a preliminary way or more defined way. From a bone perspective, a couple things come to mind. We published a paper a few years ago showing that the more fish you eat during spaceflight, the less bone you lose, and that likely ties back in with omega-3 fatty acids. We've got a study going on right now looking at the role of animal protein and potassium, which is the Pro-K experiment that I talked about, using the ratio of those two things in the diet to help mitigate bone loss. We're partnering with our European colleagues who have done experiments looking at lowering sodium intake as a way to mitigate, again, bone loss. So there's a number of things that I think we could optimize when it comes to trying to help bone the most. And then there's a number of other factors as well. Oxidative stress is still an issue that we need to look harder at. There's some potential nutrition components that we've identified that we're looking at further with relation to the vision issues during flight. So while we've learned a lot, we still have a long ways to go, but we're making progress.
James Dean, Florida Today. I think Ms. Robinson again. Are there, based on the way Dragon re-enters and splashes down compared to the shuttle's landing profile, are there any samples, any research that's considered too delicate that can't withstand return on Dragon, and so maybe you just wouldn't bother including them in down mass? And maybe Dr. Yateman, I wonder if that might be true for protein crystals.
It has been a concern in the past that the shear forces coming back down might just destroy the crystals, the fine crystals that are made. So, one interest is to potentially image some of these crystals up in space before they come back down. So that's one thing we're looking into.
Yeah, so it isn't as smooth of a ride as landing on the shuttle was, but it is enveloped by the kinds of requirements that we get for launch as well. So we do worry about it for certain sensitive samples like some proteins, but you can also pack smart and pack things up, have things padded and so forth, and take care of at least that last, it's that landing part that's plus the vibration and shaking. Those are the two kind of components that could damage things. So overall, things that can launch well can land well. There are a few exceptions of things where you think about it.
Over here.
John Walkowitz from GeoG News and Countdown Today. My question has to be about the bone loss and mass density. When you were talking about it doesn't have an effect when they return back to Earth, how long did it usually take a crew member or an astronaut to get that strength back or get that density back into the bones?
Well,
and that for anybody.
Those are really two different questions, and it gets into some of the way we image bones. But what we showed in the paper last month, partnering with the Bone Lab and with the Exercise Lab, is that you can maintain bone mineral density. There is still a question of whether or not that bone is as strong as it was before flight, because there's some biochemical changes that we picked up in the blood and urine which suggest that it's not the same as as going back to having gravity. Okay, what we're doing is the exercise is increasing bone formation, which balances out the increase in bone breakdown, which is still happening. So we don't know enough about strength, and there's studies that are being implemented right now to look at bone strength to see if we're improving that. To go back to the other part of your question, it's been estimated it takes about 2 to 3 times the length of the mission to recover the bone. So from a 6-month flight, we're looking at 12 to 18 months to get back to full bone mineral density.
NASA and the Russian Space Agency collaborate and compare data from long-duration flights from their past programs to try to come to a result when it comes to bone mass and density?
We have a long history of collaborating with the Russians, going back into the 90s with the MIR programs, and especially on the medical side, there is a lot of discussion back and forth between the programs. With the research endeavors, at least our research tends to be focused on the USOS crews, who are the ones who participate in our experiments.
In the back.
Ronnie Williams, social media. Since we're on the subject of bone, you answered a couple of my questions. What's the percentage difference versus the early space program and now, since we've been examining this, that as far as bone loss, what's the percentage difference?
Okay. If I can have the last chart. This is actually an image from the paper that we published. It may take a minute. But what we did in that paper is we looked at normalized, if you will, bone mineral density loss per month, because that scales it out by the length of the mission. And what we have here is 4 bars. On the left is the MIR. The middle 2 bars are crew members exercising with the interim resistive exercise device, the IRED, as we called it. And the reason there were 2 splits there is because the IRED bar on the right, expeditions 14 through 18, are crew members that we collected blood and urine on during flight. So that's why we broke those out. And then you can see the advanced resistive exercise device on the right. So just looking at that, the average whole body bone mineral density loss was about 0.5% in those earlier programs, compared to essentially zero, if not a little bit positive, in crew members using the ARET. And we also looked at, again, in the publication, we looked at specific regions. We looked at spine, hip, you name it, and saw the same types of trends, but there's nothing more striking than that whole body picture. Thanks.
Okay.
I didn't plant that question.
All right, that's about all the time we have. Julie, would you like to make any closing comments?
Sure. You know, so we brought to you today 4 different scientific perspectives of what's going on the space station. And just to kind of recap that, we talked about the diversity of disciplines. There's no other laboratory I know of with such international breadth and such breadth of disciplines that can do research that can't be done anywhere else. Then Dr. Yateman really talked about opportunity. With cases growing, offering new solicitations, there are opportunities far beyond what NASA's been able to offer in the past that are growing through cases management of the National Laboratory, and that's a really exciting development over the last year and continuing on. And then with Dr. Nielsen Price, you know, she's an example of new investigators who've never done work in space are getting access, finding opportunities to get the funding, and really ask basic questions, and that those experiments can happen pretty quickly. And then finally, what you saw with Dr. Smith was how our long-term investigators that get multiple flight opportunities over time can really start moving fields forward, make advances stepwise, and you really see that coming out in his work. So I hope that that gives you overall a sense of ISS as a dynamic and fully established laboratory. It's nothing like it was a year ago when people were mostly still talking about assembly. It's really full bore, full speed ahead with lots of opportunities. Thanks.
Thank you. And you can read a lot more about research aboard the International Space Station and the SpaceX CRS-1 mission that is launching to the space station by going to our website at www.nasa.gov/station. And our next televised event coming up here on NASA television is the SpaceX CRS-1 pre-launch news conference at 6 p.m. Eastern Time. And we'll see you then. Thank you very much for coming.